Hi everyone, my name is Fabien Vivier. I'm a fifth-year PhD candidate at the University of Hawaii, and today I'm going to talk about our work using drone photogrammetry to monitor the age structure and develop a growth curve for a critically endangered population of bottlenose dolphins in the Mediterranean Sea. Before we start, I'd like to acknowledge our founders and partners who are listed on this slide. So the understanding of population demographic and life history traits is essential for the effective monitoring and management of long-lived and slow-reproducing species. Recent studies showed that healthy populations usually exhibit a stable age structure over time, and that age structure is strongly related to abundance and population status. Age structure here simply reflects basically the proportion of animals at different life stages within that population. Morphometric data of cetaceans contribute to the assessment of individual and population-based reproduction, health, and demography. However, despite its many applications, obtaining accurate morphometric data on free-ranging cetaceans is rather challenging. So there is a need for improved assessment techniques which can provide a rapid indication of population status and trajectory. One way we can approach this is to use UAS photogrammetry or drone photogrammetry. Some of the advantages of drones are that they're inexpensive, they allow the sampling of large groups of animals at once, and also monitoring the demographic parameters for those populations. Drone photogrammetry has been shown to be a robust tool for baleen whales, and more recent studies show that it has a high potential for small delphinids as well. So in collaboration with Tethys and the Ionian Dolphin Project, we use drone photogrammetry to monitor a critically endangered subpopulation of bottlenose dolphins in the Gulf of Ambracia in Greece. This long-term study provides a unique opportunity to apply drone techniques to non-invasively collect body morphometrics of those dolphins and link that with the life history information such as the sex, the age, and the reproductive status for those individuals. All in all this information is going to help us understand more about the status of that subpopulation. Those animals live in a strongly affected and polluted environment with high concentrations of pesticides that have been found in various forms of life in the Gulf. And there's also increasing anthropogenic activities such as fish farming, agriculture, or sewage discharge, which is basically accelerating the eutrophication process of this environment. So in terms of data collection, we collected data for 2 years in the Gulf. Whenever we found groups of dolphins, we would deploy the drone and fly over those animals. Whenever those animals would come up to the surface, we would basically take a photo of their dorsal fin. That's going to allow us to know exactly who those animals are and access their life history information. A summary of the data that has been collected. In 2021, we spent 4 days collecting data. We measured 101 unique individuals. In 2022, we spent 6 days and measured 97 individuals. Across the 2 years, we measured 149 unique individuals with 47 repeats, including 14 calves and juveniles. So the first thing that we need to do here is to estimate the size of those dolphins. So we're using a method that we developed as part of my PhD, and that's currently in review. Basically, we showed that drone photogrammetry is very accurate when we measure dolphins in stationary or elongated form at the surface of the water when they show up their rostrum and their fluke. When we use the blowhole to dorsal fin distance to estimate total length of those dolphins when they come up to the surface to breathe, we tend to overestimate the length of those dolphins by 3.3%. So here in the Gulf of Ambracia, we started by extracting images from the videos to measure the animals in total length and measure the blowhole to dorsal fin distance when they were elongated at the surface of the water. We collected images or repeated samples for 77 individuals, and we looked at the relationship between the measured blowhole to dorsal fin distance and the total length of those animals. And basically, we looked at the relationship in 2021 and 22. It's the same, it's very strong. So that allows us basically to use linear models to estimate total length based on blowhole to dorsal fin distance for when those animals are surfacing. The second thing that we did was estimated a minimum age for those animals. And to start with, we had to establish the birthing seasonality for that subpopulation. So using calves for which the first sighting was less than 6 months from the last sighting of their mother alone, we managed to establish the birthing seasonality. We had 26 calves for which we could do that, and basically the seasonality extends from March to August with a peak in June. What we had to do then was to estimate the age of all the animals that we measured. So for the calves that were born within 6 months of that last mother alone, we established date of birth as the midpoint in that time frame. For calves that were born more than 6 months, we calculated the probability of being born within the season that they were sighted, and then for adults, based on photo ID and other photographs, we established that they were adults, and we estimated them to be at minimum 10 years old at the date of their first sighting. So if we have total length for those animals that we managed to get from blowhole to dorsal fin distances for when the animals are surfacing, and we also have a minimum estimated age, we can establish a growth curve for this subpopulation. We used 4 different growth models, and the Richards model ended up being the one fitting our data the best. The growth curve that we found is very similar to another population of bottlenose dolphins in Bunbury in Western Australia. And that subpopulation there lives in a cold environment that ranges from 14 Celsius to 23 Celsius. What they found there is that animals that were born in the summer have to grow really fast within the first year of their life to make it through the next winter. And we find the same pattern in the Gulf of Ambracia. Animals in Bunbury and in Greece also tend to grow to a maximum size of about 250 to 260 centimeters. The other thing that we looked at was the age structure of this population. So in order to determine the age structure of the subpopulation of Ambracia, we used another method that has been also developed as part of my PhD, and that's also in review right now. To do that, we basically collaborated with the Sarasota Dolphin Research Program in Florida, where we calibrated this method with basically their morphometric data that they have through their health assessment research. I'm not going to go too much into the details here, but basically we simulated drone measurements based on their morphometric data, and we tried to see how well we would perform in terms of assigning individuals to an age class based on the simulated measurements. Overall, the method is not perfect, but it showed that it was somewhat promising in order to categorize animals in specific age classes. Here in the Gulf of Ambracia, we did the same with our measurements. So basically, using the blowhole to dorsal fin distance, we estimated total length as we described, and we split the population into 3 different age classes: 0 to 2 years old for the calves, 2 to 10 years old for the juveniles, and 10 plus for the adults. Looking at the residency data, we also managed to look at what was the age structure for that subpopulation, and what we found is that using those morphometric data collected with the drone, we managed to get a very comparable age structure for that subpopulation. So the significance of this work: we first developed an age length growth curve based on drone measurements and demographic data that we combined. That is going to help us understand how those animals adapt to an environment that's under pressure. The second thing that we did was determining the age structure of this subpopulation. So while the results are still in the process and preliminary, this shows that it has a high potential of replicability for other populations that don't have as much information on. In terms of the next steps, we are going to conduct another field season next month. And for animals that we're going to manage to measure again, and that have been measured in 21, 22, and potentially 23, we are going to look at the individual growth for those individuals. So we're going to mostly focus on calves and juveniles. We are still working on refining our methods, especially in terms of aging the animals. And the last thing that we're interested in is looking at the potential of drones to detect pregnancy stage, and mostly for the females that were measured in the summer with the drone and that later were found with calves in September or October. So thank you for your attention, and then if you have any questions, feel free to ask me.